Are you there?
Yes, I can hear you.
Surajit Das, sir?
Aurojit Dash, I think he is having some
Oh,
connectivity
so
uh,
connectivity problem.
actually, he cannot
We
hear.
may start because it is 9:31.
9:30. Yeah. So, a very good morning again, and this is the second day of our 5 day long workshop of next generation big data biology. And yesterday we have learned about the metagenomic, the importance of metagenomic studies and big data biology. Uh, Last day he has given us a very good introduction. So today we are very eager to learn from him. So without wasting much time, I am inviting Mr. Shubhajit Roy to start his discourse. Mr. Roy.
Thank you, Dr. Rai. I'll be sharing my screen. So I hope this is visible to you all.
Yeah.
Yes, sir, it is visible, sir.
Okay. So good morning, all of you. And as Dr. Rai told you that yesterday I was talking about the introduction of metagenomics, and I have give you a glimpse of how expandable the science is, and it's very new to the microbiological field, and it's gaining momentum, and we are hopeful that in near future we will be able to see its progress and some monumental achievement it will gain through its milestone experiments. So before so as I have discussed that metagenomics is actually dealing with the extraction of microbial samples directly from the environment. It may be soil, it might be a marine or a riverine stream, or it might be a fecal matter or oral swab from collected from any human. So these techniques required two approaches. Either you can go for a survey, that is, you can go for a PCR based approach, that is 16S rRNA sequencing, or either you can go for next gen sequencing through whole genome shotgun sequencing. So for both the purpose, you need to isolate the genetic material from the microbes, from the environmental samples. So what is the most challenging part of metagenomics for the DNA extraction is that you will learn that the next step of DNA extraction is preparing a library. So if you are going for a PCR based amplification, or either you are going for a whole genome shotgun sequencing, depending on the type of experiment you have designed, the only limitation is that you have to choose very wisely the DNA extraction process, because what are the library preparation techniques are available, they require some specificity, some amount of purity, depending on what platform you will choose for NGS to run these samples. So some requires very short DNA fragments with pure quality. Some of them have requirement that the presence of salt can inhibit the library preparation process, or some of them have the presence of organic material can inhibit its reads and other downstream processes. So DNA extraction is a very essential part of this metagenomic uh, application. So the completion of Human Genome Project has heralded one of the major landmark events in science. So the human genome contains the blueprint for human development, the maintenance, and ultimately provide the means to understand how human cellular and molecular processes, both health and diseases, uh, And the genome is full complement of DNA from an organism and carries the information required to specify the structure of every protein the cell can produce. The realization that the DNA lies behind of all the cell's activity that ultimately leads to its development is also what we know and learned about learned in molecular biology. So we need to identify how it is important for us for performing a metagenomic analysis, the DNA extraction would be. Because for the metagenomics, we are not concerned about one or two microorganisms. We are isolating it as a community. So there will be presence of many microbes and other substances. So we need to have a universal approach of isolating genomic DNA from all the organisms. It's very easy for if you are dealing with a blood sample or a human tissue sample or any other tissue samples, maybe isolated from rat or any organism or even plant. All the cells are similar. All the cells have their own similar type of genetic material. So with an universal isolation technique, you can just extract the DNA from the cells and go for its downstream analysis. But think of the diversity it has in a microbial samples in the isolated from uh, 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 an agricultural field, or maybe if you are dealing with microbes isolated from human anatomical locations. So you will have the human cells which are eukaryotic in nature, you will have prokaryotes, you will have fungi, you have nematodes, all over, all things will be influencing your data. So we need to have an universal approach of extracting the DNA from all types of microorganisms or microbes present in that sample. So Friedrich Michener, this Swiss biochemist, first introduced how to isolate nucleic acid. It was in 1869. Since then, been uh, we have been introduced to how deal with the nucleic acids. So for the eukaryotic cells, as you can see in these cartoons, it has a well-defined nucleus where the DNA resides. Along with it has other cellular organelles like mitochondria, vacuoles, ribosomes, and a cell. Of course, they are surrounded by a cell wall, which is composed of lipids and proteins. The ribosomes and other intracellular organelles will be present there. So isolating an eukaryotic cell, we need to extract the nucleus first. And then by lysing the nucleus, you will be able to identify, you will be able to extract the genetic material. But for prokaryotes, what we have that the genetic material is not located in a well-defined nucleus. Either you have a plasmid, which is a small copy of the genomic DNA, and of course you will have the genetic material present, and it is not defined in a nucleus. That's why they are called prokaryotes. So the prokaryotic cells have rigid cell walls. Some have thick, depending on whether they are gram negative or gram positive. The cell wall components will be have thickened cell wall or very thin cell wall, and that's how you select how to rupture this cell membrane and isolate the genetic material out of this debris. So why it is important, or what is the importance of DNA extraction methods for metagenomic? It is an essential step in all cultivation dependent approaches to characterize the microbial diversity. including that has been associated with the human body. So because we are not going to culture all the microbes. So this metagenomic approach, I have in the last discussion, I have told you that we are not going to culture it. We are directly extracting the genetic material from the environment. So when we are isolating such microbial diversity, along with that, depending on the sample where we are extracting it, if it is on the human body, the human cells are also going to come along with the microbial genetic material. The fundamental challenge in using this approach has been to isolate a DNA that is representative of the microbial community sampled. As microbiome analysis becomes applicable to an increasing number of scientific area, a streamlined process for efficiently extracting DNA to generate 16S rRNA gene amplicon or shotgun metagenomic sequencing data from a range of environmental sample types is increasingly important. The DNA extraction is the largest source of experimental variability in 16S rRNA sequencing analysis. Because I will explain in the next slide about this one. And the complete bacterial lysis and DNA purification can be particularly time consuming, representing a significant bottleneck or restriction for the high throughput analysis. So these are the challenges what I was talking about. Extraction biases. See, when you are isolating samples from, take for example, you are isolating it from soil. The soil will have multiple microorganisms. It's not a single type of microorganisms, and they will be present in different quantities. So if you have selected any isolation technique that only can isolate the genetic material from the cells which have thin cell wall or soft cell wall, they can you will be able to get only, and when you will be at the end of your metagenomic study, you will have no idea whether there are any bacterial or microorganisms were present, though from which you haven't isolated the genetic material. So if you have so this extraction bias can affect your data analysis. On the other hand, as one of the part of metagenomic study is 16S rRNA sequencing, so this will deal with PCR amplification, and we know there are many things present which can affect your PCR amplification. So if your sample is contaminated with proteins, humic acids, polysaccharides, it can pose a huge challenge for library preparation as well as amplification of the targeted region. So only if you have a small quantity of any particular species, that thing will not be, if your PCR is inhibited, those genetic material will not be amplified, and you will not get the, you will not be able to identify those organisms in your sample. So there are several steps by which you can extract DNA from the organisms. So first one will be then the DNA extractions consist of 4 basic steps. First of all, preparation of the cell extract, that is lysis, and you can perform lysis on a mechanical way or enzymatic or chemical way. Rather, it is preferable that if you are isolating microbial populations from different environmental sources, to apply both mechanical and enzymatic or chemical approaches to completely disrupt the cells and extract the genetic material. For the purification of the DNAs of the cell extract is very important, because if any of the materials are present which can inhibit your PCR or subsequent downstream analysis, it will be a huge loss, because at least for NGS or shotgun sequencing, you have to pay a lot amount of money compared to the 16S RNA amplification. So for the purification of the DNA extract, either you can go for adsorption or you can go for a precipitation. You need to analyze the perfect concentration of the DNA samples, because a certain amount of DNA samples are required for library preparation. And of course, the measuring the purity of DNA, you can use a nanodrop or a spectrophotometer for identifying the quality, or if there is any contamination present in that isolated DNA sample. So for the isolation of the environmental DNA sample, if you are doing an in situ extraction, the advantage is high yield of DNA and with low extraction bias. But for the ex situ one, a very large DNA fragments up to 20 or 500 kb fragment will be generated, and less contamination of the non-microbial and free soil DNA will be there. So disadvantage is that DNA is shared into small species, that is 1 kb to 50 kb in length, and with more contamination. Where the ex situ extraction is the low end of DNA with high extraction bias. The purpose of these two, in situ or ex situ extraction, will be different if you are isolating it from the soil. That if you need to clone it a small plasmid lambda vector for one gene or short gene cluster expression test only, then you only can go for short in situ extraction. But for ex situ extraction, you need to clone it into a larger vectors, like who can handle a larger insert, like BAC or bacterial artificial chromosome, or cosmid vector for a huge chunk of the genetic material can be cloned into that. So for the cell disruption, you can go for two different type of approach. One is mechanical and one is non-mechanical. Majority of us has been introduced to the non-mechanical way of cell disruption, of lysis of the cell, that is either acid or alkali based enzymatic degradation, either if you are applying a proteinase K, or if you are doing lysozyme technique for disrupting the cells, then these are called a non-mechanical way of doing that. And osmotic shock, if you are doing a create a solution which can disrupt the solution, enter into that and disrupt the solution, disrupt the cell, you will be able to get the genetic material from that. For mechanical way of disrupting the cell, there are two possible ways. There's solid shear and liquid shear. For this solid shearing, you can use a bead beating technique or a press, or for the liquid shearing, a high pressure homogenization and ultrasonications. And other techniques are autoclave, lyophilization, and microwave. So let me remind you, these all techniques which I have discussed here are not meant for DNA extraction only. So I am only discussing the cell disruption techniques which can be applied to rupture the cells. So you have to choose wisely that what are the things you require for your proper and pure isolation of DNA depending on the experiments. So the cell itself is surrounded by cell membrane, which contains a lipid. As you can see in this image, that it has two long chain attached and a phospholipid group, and they form a bilayer. So when mixed with membranes, the hydrophobic ends, so they form ends of the detergent bind to the hydro. So if you are applying SDS or CTAB or other enzyme or other detergents to rupture these phospholipid molecules, they can form a micelle-like structures and forming these are the hydrophobic tails, as you can see here, and these are the hydrophilic end. So these heads will orient themselves towards the water molecule and hide their hydrophobic tails and forming a detergent micelle. So using this technique, we can disrupt the cells, and majority of the lysis buffer, you will find there is a detergent present. So one of the famous DNA extraction MAKAUT procedure, which we have here, is the phenol chloroform isoamyl alcohol method of DNA extraction. So for that, we need to lyse the cells. From the lysis buffer contains tris HCl, EDTA, which is a chelating agent, NaCl, magnesium chloride, and sodium dodecyl sulfate. And this lysis buffer have a pH of 8. So what this does is that EDTA chelates the calcium and other divalent metals which are required for the cell membrane integrity. And SDS removes the lipid content of the cells, and after vortexing and centrifugation, all the cellular debris along with the DNA will precipitate at the bottom of the Eppendorf tube if you are using it at the end of as a pellet. And then you need to add saturated phenol. So saturated phenol means it has been buffered with tris HCl. So phenol itself is non-polar. So when you try, and you know that DNA is genetic material, it's negatively charged because of the phosphate backbone. And when the DNA polar negatively charged phosphate group will tend to dissolve in the tris saturated phenolic layer, instead of if you have dissolved it in water, it will not, the DNA will not dissolve in it. So when you saturate that along with the phenol chloroform and isoamyl alcohol treatment, what you will find that DNA will be present in the aqueous layer, while lipids will dissolve in chloroform layer as an organic material at the bottom of the tube. And between these two interface, protein will form a layer. So I will show you the exact image in the next slide, how it happens. So as you can understand why the protein is settling in between these polar and organic layer, because these proteins have both polar and non-polar amino acids. So they tend to settle between these two layers. So after collecting, you can these aqueous layer, you can repeat the step. Isoamyl alcohol, which is present in very small quantity, as you can see, that it prevents the foaming of phenol. So if you agitate phenol, it will tend to form frothing, and the presence of isoamyl alcohol will prevent that to happen. So once you have collected this material, aqueous layer of DNA, though some trace amount of proteins will be present there. So you need to precipitate it using ethanol and sodium acetate. Now, as we are dealing with an aqueous solution, of course, there will be presence of water. So water have a high dielectric constant, about 80, and it tends, that's why it's an universal diluent. So when you add ethanol, so it tends to reduce its dielectric constant, and what happens, so the water molecules form a hydrogen bond along with the ethanol, and sodium binds to the DNA along with the phosphate backbone, and then tends to precipitate it at the bottom. Once it has precipitated, you can centrifuge it and wash it with 70% alcohol where the water proportion present in the 70% alcohol will tend to dissolve away the salt present in the DNA extracted, and you will get a extracted DNA. So this is a conventional technique we majorly use for isolation of the DNA in most of the molecular biology lab. If you go for any genetic lab or a molecular biology lab where they regularly isolate DNA as a genetic material, you will find they are mostly following these techniques. So there are other variations too, where they use sucrose solution for lysis of the cells. Some of them use other buffers, and but mostly the techniques which are present has been used are use the similar principle. So this is what I was talking about. So DNA will be present in this aqueous layer. Protein has sedimented between these two layers, and as you can see, the lipids deposited in an organic layer between this beneath this protein layer. So how as I have told you that several other techniques are there. One of them is a salting out technique, and as you have already seen how we have used ethanol and sodium acetate. So increasing the salt concentration in moderate amount will initially help to increase the solubility to some extent. But once it has reached a maximum quantity, what will happen? They will tend to form interactions between them, leaving behind. So if this is the material you want to extract, and these are the salt. So initially they are forming hydrogen bonds and helping these solution to homogeneously mix in to form a solution. But once you have reached a critical concentration, instead of forming more dissolving the material, it will tend to precipitate it. As this we called a salting out technique. So another thing we use commonly, and you will be able to see that majority of those who are performing metagenomic and analysis are mostly concerned with the purity of the DNA. They follow a column-based DNA extraction technique. So they provide you, it comes in a kit, and they provide you this kind of a tube where you have in this region you have a silica membrane. So this is an adsorption based technique where if you after lysing, you add the lysed solution onto this column, and after spinning, all the materials will pass through this column except the DNA will be bind to this silica membrane. So as you can see how this chemistry works, this is when it is in nascent stage. So it forms the water molecules occupies this silicate group, and when you add sodium in the presence of salt concentration, it forms a brief interaction with the DNA molecule present in the solution. After doing this, if you go for repeated spinning, you will left behind with the DNA only bound to its silica membrane, and all the other debris, the proteins and other things will be passed through the solution. Then using different salt concentration and alcohol washing, you will be able to wash whatever left debris are left in this column. Only the DNA will be binding to this membrane. Now, when you again add water molecule, this will go back again into its native state, leaving behind the DNA as an eluted in the solution. So once you spin that, you will get the purified DNA fragment. So when you will be using the kits, you will find that different companies have different compositions of these salt washing buffers which are provided, and it can affect the quantity of the eluted buffer, eluted DNA. And this is mostly purified DNA fragments, and you can go for subsequent downstream analysis. So this is a basis how we use this technique. So this is the membrane I was talking about. So lots of free protons and neutralize the silica. So when we add a high salt concentration, mostly they are provided with guanidinium salt. So these we call is a chaotropic agent. So it loosens up the water molecule coat from the surrounding DNA and denatures the DNA pool and helps to shield the negatively charged, so that, as you can see here in this previous slide, that the silicate group was previously occupied by water molecules, and DNA, which was generally which is of course negatively charged because of the phosphate backbone, is now showing positively charged because of the presence of sodium ions. So similarly, this what happens, it shields the DNA with a positively charged, and it can bind with the silica and forming extensive bonding. And when you add water, it will be eluted into the tube. So what is a chaotropic agent? So this substance which disrupts the three-dimensional structure of the macromolecules such as protein, DNA, RNA, and the chaotropic agents interfere with the stabilization intermolecule. So if it is a DNA, so it will interact with the water molecule and try to become stable. So uh, in high salt concentration, so it tends to bind with the silica, and in low ionic strength, when you adding water, so it will be eluted from the charged bed. So these chaotropic agents interfere with the molecular interactions, which are by non-covalent forces like hydrogen bonds, van der Waals forces, and hydrophobic effects. So apart from guanidinium salt, there are many other chaotropic agents are present, like sodium iodide, guanidinium hydrochloride, urea, and you can use it in compositions also, depending on what type of kit you're using. Another new technique has been used in the preparation of uh, the isolation of genetic material, is a magnetic base isolation technique. So these magnetic beads are actually paramagnetic beads which are coated in silica. So the techniques is similar to the membrane based isolation technique, only is that when we apply these beads, it selectively binds to the genetic material, and on the when we place it on the magnet, it will tend to precipitate at the bottom because of the effect of this magnetic field, because these are paramagnetic beads and only show their magnetic property when they're exposed to their magnetic fields, and leaving behind the debris and whatever unwanted materials will be present in that solution. So you can pipette that thing out, and after that you can add water or any elution buffer which will remove the beads from your isolated DNA, and you can elute the DNA from the beads. So this kind of technique uses SPRI technique, or solid phase reversible immobilization technique, where you can, as you can see, that silica based magnetic beads are immobilizing the DNA in the presence of these magnets, and they're reversibly binding, because when you are adding the elution buffer, the magnets are again removed from the solution or the isolated DNA. So this yields a very pure quality of DNA, and it's very essential for if you are if you have the idea of performing metagenomic assays. This could be an interesting to you. So of course, after isolation of the genetic material, it is very important to make you understand what are the what is the purity of the genetic material you have isolated. So either you can go for a nanodrop or go for spectrophotometer. The principle is that nucleic acid will be able to absorb the uh, absorb the light at our wavelength at our 260 nanometer, and in 280 nanometers the protein will be absorbing. So if you have it's a basic thumb rule that 260 by 280 ratio, if you have 1.8, then probably your DNA is pure. And if 260 by 230 is for the organic materials. So you will have a higher ratio of 1.8. So approximately when you have a pure form of RNA, then your this 260 to 80 ratio will be around 2. So I personally prefer, along with whatever, even if you are performing uh, uh, a spectrophotometer based or a nanodrop based purity quantification, always go for agarose gel electrophoresis to identify whether there are any DNA present or not. So because sometimes this can give you a false positive data, but it is very important to analyze it on the agarose gel and visualize it using ethidium bromide or phenol red kind of identifier. So this I will explain, give you an introduction of how to isolate DNA from a complex sample. By complex samples, I tend to mean stool or soil, because they have from the other environmental samples, like if you're isolating from the water, you can just dry out the water and your or you can filter it through the membrane. I will get the microbial populations. But for the stool or soil, you have not only for the stool, we will have all the things, all the genetic material what you have ate, and of course the host genetic material will be present there. And from the soil, it will have debris, dead plants. If you're collecting from agricultural field, you will have the DNA isolated, DNA extracted from worms and other unwanted materials. So we need to have a very good technique of isolating the genetic material from this amount or the sample. So first, to prepare the lysate, we use a bead beating technique. So this is in composition with mechanical, chemical, along with the heat treatment. So they have small beads and rigorously vortex or shake it, which disrupts the cell, and you heat it in 65 degrees centigrade, along with the chemicals which has been provided by the kit, will disrupt the cell wall, and you will be able to get the genetic material exposed to the solution. You need to add some cleanup buffers. These cleanup buffers remove some downstream inhibitors. Like I have already told you that humic acids present in the soil could be an inhibitor of PCR if you are trying to go for shotgun sequencing or 16S RNA sequencing. So for bilirubin, bile, humic acids, digested food for the stools, these things could be inhibitors, and cleanup buffer removes these downstream inhibitors from the sample. Then after repeated centrifugation, you need to bind it to the DNA column. As I have explained this column technique, how to isolate the genetic material from it. And after centrifugations, you will be washing it to remove any remnant of unwanted materials, and centrifugation, and finally the elution, when you add the elution buffer, or mostly it's water, RNAs or DNA is free water, and it can elute your genetic or interested genetic material will be eluted in the tube, and you can go for your library preparation, or then this sequencing. So let me remind you that when you are isolating from stool, you are any from any host, then you will have the genetic material not only from isolated from the bacterial and microbial population, but along with the host DNA. So when you will assemble it, you will have the contigs, and you can refer to the reference sequences which has previously been uploaded to the database, and then segregate which DNA was from your host material and which was from the microbial DNA population. So this is what I was going to discuss today. So if you have any questions, you are welcome.
For participants, if you have any questions, please post it in the chat box or in the question answer section.
Thank you, Mr. Subhrajit,
Thank you.
For your illuminating lecture. My question is, please to mention the troubleshootings during this process of DNA or RNA extraction generally faced by the experimenters.
Right. So the major problem is either you have an extraction bias, and the major two challenges are there. So if you have from the sequencing or if you have from the cultures, you have an idea from that it is recommended that you repeatedly sample from one site. So say, for example, you are isolating microbial DNA from one site. So it is expected that you will be repeating your DNA extraction from that site 1 or 2 times or 3 times. So if you are having a trouble that within 2 or 3 days, you will not be able to get the same amount of data, because it is not expected that within a brief amount of time the microbial population will change, especially if you are, say for example, you are isolating it from the acid mines. So their microbes grow very slowly. So if you are getting that microbial populations are not get isolated properly, then probably you have problem with your extraction. You need to select other extraction technique where you can uniformly identify your microbial population. There are several articles available on PubMed where they have compared different different isolation techniques for the microbial populations, and compared which one will be better suited for different types of sample. And for the genetic, for any kind of genetic material isolation, the common problem is the inhibitors which are present. So it is better to identify if your sample is having any contamination with the microbes, so you can repeat the steps, or you can go for a bead-based magnetic bead-based isolation, where you will get more purified form of DNA for your extraction. And for the if you are selecting chloroform or chloroform isoamyl based alcohol based technique, so the isolation of the genetic material, sometimes the presence of phenol could be very can affect your experimental design. So it makes sure that you don't have any phenols left over in your solution, and even ethanol can inhibit the PCR. So after washing with 70% alcohol, it is recommended that you dry the sample and then elute in TE buffer for long-term storage.
Okay. Another question is, suppose a sample of night soil is taken.
Mhm.
So, for an experimenter, how many times that experiment of DNA extraction should be repeated to get an appreciable amount of DNA?
Say from initially, if you are isolating from the soil sample, approximately 2 grams of soil DNA is enough for isolating DNA from the soil. So, and if it's for the purpose of getting, and if you have a previous idea of what are the genetic materials could be present there, then you can compare and streamline your techniques and review your working pipeline for that.
So, any question from students' end? Any question?
Participants, if you have any questions or you have any query, then please post it in the chat box or in the question answer section. We are about to end the today's session. So if you have any questions, please post it in the chat box.
I think no question is there, no query is there. So, Dr. Sharojit Dash, do you have any question or anything to add? So,
Sarojit
thank
sir,
you.